So the first speaker of this session is Dr. Zhang from the University of Cambridge. She is the 2020 winner of the RSC Felix Biotechnology Medal. We are very delighted that Dr. Zhang has chosen this meeting to receive her award. So prior to her talk, Professor Stephen Harding, chair of the RSC Felix Franks Biotechnology Medal Awards Committee, will award the medal to Dr. Zhang. So let us welcome Professor Harding and Dr. Zhang. Before we start this award, please unmute your click the button that your voice come out. So when this award is in the end of our award, everyone can hear your claps. Okay. So please go ahead, Professor Harding.
Okay, thank you, Professor Wang Mei, and it's a great honor for me to be presenting this year's the 2020 Felix Franks Biotechnology Medal on behalf of the Royal Society of Chemistry, an award which is given to early career scientists who've done outstanding work in the field of chemical biology. Felix Franks was formerly a professor of botany here at the University of, and is famous for his multi-volume treatise on water. That's why he's been known as Water Franks, but also in the fields of freezing and carbohydrate biotechnology. And the award is to commemorate those achievements, and the award is sponsored by the Felix Franks family. This is the second year of the award, and we were overwhelmed by the quality of the nominations, but one nomination stood out above the others, and that was Jenny for a great work in the field of Photocurrent. So it's a great honor for me to hand over the 2020 Felix Franks Biotechnology Medal to Jenny. And,
yeah, congratulations.
In true keep with the Olympic Games tradition, we have also the flowers for Jenny. Well done, Jenny. And the Felix Franks Medal lecture will be photosynthesis on an electrode. But first, Jenny.
Great. Thank you so much, Stephen. Thank you. I'm extremely appreciative because Stephen came all the way from Nottingham to give me the award in person. So perhaps I can start my talk. Let me just try and share my screen. Is that sharing? Sorry, I can't see where it is start sharing.
You should have start sharing at the bottom of your screen.
Yes, sorry, I'm just having a look. Okay, there we go. Yes, so I like to thank...
Sorry, Jenny, nothing's sharing. Oh, it's just coming up now for us. Okay? There you go. Perfect. And that's fine. Sorry, it was just a bit slow.
Yeah, that's okay. Yep, great. Thank you. So yes, thank you again, Stephen, for the lovely introduction. Really, really delighted by this award. Also, thank you very much to the Felix Franks Award Committee using me, and also to the family for sponsoring this award. I also want to thank the organizers, RSC, as well as the chairs, for letting me have this opportunity to receive this award here, as well as to share with you some of my work. So I'd like to dedicate this particular talk to Felix, and I've got his quote here, which is in one of his many volumes. Okay, interest in water in all its forms will continue for a long time to come. It will spread over a large number of disciplines and technologies, and how correct he is, because he's been applying his knowledge of water towards chemical research. But of course, we know, especially from our plenary today, that water is incredibly important in sustainable energy development. A lot of us here, even in this session, will be trying to manipulate water in several ways, splitting it into so that we can use the electrons for various fuels. And I'm no different. I'll be using biological machinery to do that function. So without further ado, let me give you the motivation to our research. So here I've just got a snapshot of how we're presently using our energy today in UK. So this is actually collected from last year. And as you can see, the UK's total electricity generation in the past few years, in last year, was formed still significantly by fossil fuels, but pleasingly, renewable energy is appearing. And you can see here, bioenergy in particular is becoming quite prominent. It's at 12.6% contribution to electricity generation, and that actually reflects also globally. So about 13 to 14% of our total energy global consumption is in the form of bioenergy. So what is bioenergy precisely? So actually, bioenergy actually originates from photosynthesis. So here I have an example of crop plants, and how we basically generate, how they are generated is through the process of photosynthesis. So let me just give you a very brief overview of what that process actually is. So if we zoom in on the photosynthetic machinery of plants, what we the organelles called thylakoids is the photosynthetic electron transport chain. So everything starts with photosystem 2, which is a benchmark catalyst in its own right for water oxidation. Its whole role is consists of absorbing sunlight and then using that energy to extract electrons from water. And those electrons are passed down the chain. It's absorbed photosystem 1, where the electrons are further energized by more sunlight, and those energized electrons are picked up by a shuttle called ferredoxin. Ferredoxin shuttles it to different areas of the cell depending on the needs of the cell. And one of those pathways is, of course, CO2 fixation in the Kelvin cycle, where we generate biomass. So bioenergy is essentially the combustion of biomass for electricity generation or for fermentation of this biomass so that we get biofuels. So what I want you to get out of this process is that parts of this pathway are extremely highly efficient. So for example, 2, as I mentioned, is a benchmark catalyst. Both photosystem 2 and 1 are incredibly efficient at absorbing light and turning that into charge. However, overall, as a process, biomass formation is incredibly inefficient. As you can see here, this is typical efficiencies for crop plants. And so if we think about the pros and the cons of bioenergy, okay, pros are that it's not fossil fuels, and it's low tech, so it's readily available, accessible, and obviously very scalable. However, it involves combustion, and so it is liberating pollutants. It uses up land, which leads to other problems, and as I mentioned, incredibly inefficient. If we think about what happens to the biomass before it's turned into electricity, it still needs to be dried, transported, combusted. So overall, the energy conversion efficiency is incredibly low. However, it's still very important, as I mentioned. So what my research and my group's research was really interested in is trying to think about how can we use photosynthesis in another way to give you a different type of bioenergy. So for example, can we take electrons, so rewire photosynthesis in a way where we can take electrons from an early point, collect it using electron collector, like an electrode, and then we can connect that electrode in a fuel cell configuration, for example, to generate electricity, perhaps to perform electrolysis, to produce other fuels. And actually, this particular pathway in photosynthesis has been predicted to be able power at overall efficiency around 3%. So that's an optimistic, it's not the maximal, it's just an optimistic prediction, and it's made by this reference here. However, if we can think about integrating this into a biohybrid device, so a tandem biohybrid device, which I'll talk a little bit about later, then we can even improve on this efficiency because we can introduce artificial light absorbers to help boost the amount of light that we can absorb using these type of natural systems. And so this sounds really nice and good, and a good idea, at least to us. However, it's not as easy as I made it sound. So if we think about interfacing cells to electrodes, well, first of all, we don't need to use plants. We can use photosynthetic bacteria, so cyanobacteria, which is actually the most abundant life source on Earth. So it's going to be very scalable. However, it's very difficult to enhance this pathway or to make this pathway so to collect electrons from this pathway so that we can use such a high efficiency. And that is because the interface is incredibly complex. So our approach is really starting from a simpler model system. So by connecting and examining how we can wire photosynthetic proteins to electrodes first, and then applying lessons from that to more complex living systems. So we can extract electrons from proteins while it's still being repaired inside cells. So I'm going to talk a little bit about what we've done here, the lessons we've gained, and then I'm going to talk about how we applied it to here and some of our latest results. Okay, so we're going to start simple. So this is Photosynthesis, which I already described. Benchmark water oxidation catalyst, and how it works is simply it has a lot of pigments, absorbs light, it channels those light energy to a reaction center. Catalysis of water oxidation occurs in the manganese calcium cluster, which was mentioned earlier by Harry, and these electrons are basically passed along to the other side of the protein, and if we have a suitable electron collector there, electrons will be collected. And so we can measure the collection of these electrons using a very simple electrochemical technique, photoelectrochemistry. Okay, so this is when we have immobilized photosystems onto electrodes. And what you see here is I applied different energy levels onto the electrode, and then we shine light on it and we turn off the light. And what you see here are these photocurrents, and the magnitude of the photocurrent denotes, well, it represents how, for example, how active the photosystem is. It represents how the quality of the wiring at the interface, and also can represent how many of the catalysts we have. So what is the reaction flux at the surface of the electrodes? And so you can see an example here. We can boost the current by having a more active photosystem from a more active species, and which you can clearly see by the higher photocurrent here. And so what we're really interested in this talk is boosting this photocurrent so that we can use it for biotechnology. And so one I said is to use more active catalysts, but also you can try and increase the quality of the wiring. And we've done lots of work to understand how we can wire. So I won't have time to go through it, but if interested, there's some reference here. But basically, you can use electron shuttles, you can use redox polymers, and that bypasses the need to really orient the photosystem directly in a certain position on the electrode. Now, so the wiring is very important, but what we found was even more important is actually the electrode design. And this is just a history of the photocurrent output over the years from photosystem 2 on electrodes. And you can see that for many years, there has been pretty much a plateau until we really started to play with the electrodes. And when we started to not only roughen up the electrodes, but introduce these hierarchical inverse opal structures to using indium tin oxide, which is a very common conductive semiconductor metal oxide material, which has very favorable light properties, is very hydrophilic. And by making these kind of very conductive sponges, we're able to soak up a lot of enzymes and biocatalysts and hence enhance the reaction flux at the surface of the electrodes. And we get photocurrents that's up to 3 orders of magnitude higher than previous. And so you can see that this is a lesson that we have gained. So using these type of platforms and with these type of photocurrents, we're able now to put them into prototype devices. So here we've basically wired photosystem 2 to hydrogenase to do overall water splitting. And in our first prototype device, and this was done in my postdoc years actually, when with a PhD student who led this this work, we found that there were some pretty, the result was a pretty moderate light to hydrogen conversion efficiency, but we found that we needed to input some energy because there was a mismatch between the photosystem 2 and the hydrogenase. However, this could be overcome using tandem bihybrid devices, or a tandem bihybrid approach, where you basically integrate artificial light harvesters in any devices. So it could be, for example, in one case, we use another semiconductor to on the side of the hydrogen reduction, so that we can have complementary light absorption to increase the energetics that's of the electrons that's coming out of photosystem 2. And so in the end, we could have many bias-free solar splitting devices or novel CO2 reduction devices, which was really pleasing. And this really opened up the field, what we called semi-artificial photosynthesis, which is basically another way of saying it's a bihybrid device for solar energy conversion. So this is all well and good. These devices were really interesting. We learned lots of lessons from it. But unfortunately, we were always fundamentally limited by their. They lasted no more than an hour. That's because their enzymes are fragile and they break down when they're not in their native in vivo environment. And so we really turned to looking at living systems. Actually, that would be much more convenient. We wouldn't need to purify them. Can we actually wire into the enzymes whilst they're being repaired inside of the cells? And so this is when we started studies with cyanobacteria, so the photosynthetic bacteria. And here we use a model species, Synechocystis 6803. And so first of all, what we need to do photoelectrochemistry on them is to redesign the electrode. So we enlarged all the pore sizes of the previous structure that we made for the enzymes, because obviously the cells are much larger. Okay, and then we were successful in being able to absorb a lot of the cells into our structure and being able to have very reproducible biophotoelectrochemical outputs. And you can see an example here where I compare just photosystem, purified photosystem with cells on electrode. And you can see, well, there's lots of interesting differences. I did the exact same chronoamperometry analysis as well. You see lots of interesting differences. But for the sake of this talk, I'm going to focus really on the magnitude. And you can see that it's much smaller in this case. That's expected because now the catalysts are embedded on the thylakoids, and we are trying to extract electrons that are buried inside of these membrane structures. And so obviously the photocurrent has decreased. But what was interesting though is, as we expected, the longevity of the photocurrent response has significantly increased. So here we have isolated photosystem 2, and I should walk through you on this. We have turnover number, which is basically the number of products produced by per catalyst over time. Light on and off to simulate day and night and over 5 days. And you can see here with isolated systems, photosystem 2 with a mediator, okay, very fast accumulation of products, but then it dies. And then when we have living systems, we have very slow accumulation of products, but it lasts and lasts and it does not die. Now, what is really fascinating is that we can manipulate the extent, the rate of the catalysis by basically introducing mediators to our system. In this first study, we basically used a very harsh mediator condition. We were able to really significantly increase the rate, but then we killed the cells. So obviously there's lots of room for improvement there. So what I guess I would like to summarize from that is that we have now began to really look at the photoelectrochemistry of cyanobacterial cells and analytically pick it apart. And you can see that over the years, okay, so this is the photocurrent output over the years, it has really plateaued attempts to bioengineer the pathway to increase the flux of electron going out, and that has not been very successful. Although there has been significant improvements, it hasn't been able to meet the predicted numbers that we were trying to achieve. Now, when we started boosting and redesigning the electrode interface and trying to lift different mediators as well, combined with scaffolds, we're really able to lift that. But we really want to get to this point here, which is our goal, because this is the predicted area, and this is the area where it starts to become competitive against current bioenergy technologies. So you can see we're nowhere close at this point. So at this point, there's many things we should try. And because of the fact that we have found that bioengineering has not helped significantly, we know now that the bottleneck is likely to be at the interface and at the abiotic component. So there's so many things we can explore. And I'm not going to have time to go through all these efforts. And in fact, I'm going to only talk a little bit about the mediators, just in the interest of time, and then I'll focus more on the structure. So we just recently had a study out in ChemSci about how we've been trying to understand what makes better electromediators, because as you remember from my previous slide, we're able to increase the rate of photocatalysis using mediators, but we were also unsuccessful in keeping the cells alive. So in this study, basically, we went into comparing different mediators with different midpoint potentials to see whether we can harvest electrons from different parts of the photosynthetic system. And then we tried to see what factors govern the longevity of the mediation. And we were quite successful in coming away with some very valuable lessons. And there's lots of work to be done, but we came away with lots of conclusions just to cut a long story short, I guess I can just tell you that the CPQ is by far still the front runner, and we're now starting to understand how it meets. Okay, so I've done some work on the mediators, but let me tell you about our work on the structures. So, so far, our attempts to really boost the structures of the electrodes have been focusing on increasing the electroactive surface area so that we can increase the number of catalysts to increase chemical flux or chemical reaction flux. However, as Harry has mentioned in his plenary as well, people doing photoelectrochemistry usually use other simpler geometry, so pillars. And that's because we really also should care about the light flux. Now, there's so many considerations here in terms of width, height, etc. Where do we even begin? And so we really started to use a different technology, a newer technology, I guess, to help us answer these questions. And so Xiaolong here, he is a postdoc in my group, and he does lots of 3D printing. So he's been investigating a technique called aerosol jet printing, where he's able to fabricate large libraries of micropillars very systematically and robustly. And so you can see here that he can systematically create micropillar electrodes with different heights. But not only that, he found a way to introduce micro roughness to the pillar. And this will be important because it not only roughens up and introduces extra electroactive surface area, but as you see, it's also very important for light absorption. So you can see here, we have, with comparing 3 different pillars in this study, electrodes. And in this particular test, we're looking at how the electrode architectures handle light. So on the left in the black is transmission of light, and this is reflectance of light by the structures. When we are comparing these 3 architectures of the same height, so 350 microns, we can see that the inverse opal ITO, so the porous electrodes, give much poorer transmittance as well as reflectance. On the other hand, we have the micropillars, which basically increase in reflectance and basically exhibit very favorable light transmission and reflectance abilities. And in particular, the branch pillars, which you can see here, which maybe it's a little bit hard to see, but these are what the dark red line is. That's the reflectance, a high reflectance is from the branched structures. When we compare that against electroactive surface area, okay, so with the same height, out of the electrodes with the same height, inverse opal ITO gives much higher surface area compared to micropillars, but we can overcome that with the micropillars by increasing their height, which is very intuitive. Now we loaded the electrodes with cells, and what you can see is change when we have cells on the electrodes in terms of transmission and reflectance. And that's because, well, basically, most of the light has already been absorbed by the electrode in the first instance. Now, in the cases of the micropillars, what we see is a very significant change, and that's because the third component of light handling, which is absorption, has taken place here. The cells on the electrodes have absorbed the light, and we found that actually about 30 to 40% of the light has been now absorbed by the cells used for photosynthesis, so for photocatalysis, which is a very good sign. And here we have a visual representation of how the cells are loaded on our electrodes. And basically from the fluorescence microscopy, we found that the ITO inverse opal actually had much higher loadings of cells compared to the other two. Now, in summary, what does that give us? So here are the photocurrent with and without mediator. To summarize everything, what we found was that even though with the inverse opal ITOs, okay, it has much higher loading and much higher surface area, it gave rise to the highest output. And in fact, it was the branch pillars that gave rise to the highest output. And that's because it was able to balance light transmission properties with high surface area. And this is very much a combination due to the pillar structures as well as a micron structure. So the hierarchical structure and how that interplays is extremely important for balancing the flux of both electron transfer and light energy transfer across the biointerface. And we were able to also do some statistical analysis on our big library of electrodes, because now we can with such a big library. And what we found, you can see here, this is the output, the photocurrent output from the different height of pillars. We found that there was a limitation at 800 microns in height, and that's probably where we stopped fabricating very stable electrode structures. So by doing a statistical analysis of this, we found that photocurrent density was very much correlated to the pillar height again, which is expected, but also to the light reflection properties, which is enhanced by the micron pillar structures. So where does this all lead us? So we were here about 2 years ago, and now we are way ahead. So now we're very close to the goal that we want. And this is extremely pleasing because it means that actually the goal is in reach. This is not going to be the final structure or the material that we'll be left with. We're still going forward with more investigations, but we have so many clues and lessons to go forward with. We're very optimistic about the future in this particular area. Okay, so hopefully what I was able to do was to give you a summary about some of the work we've been doing with biohybrids and how we've been investigating them. And we're really trying to optimize the stability and the efficiency at the same time with this work. Well, I haven't talked anything about versatility, and that's because I've been really giving you, a very simple example of how we can hijack into electron transfer into living systems. So as easy as we can basically hijack into the auto-oxidation pathway, we can think about how we can hijack into other pathways for reduction as well. And so that's really where fuel formation also comes through. And we can use the lessons drawn here to apply to other microorganisms. And of course, the applications of these type of biohybrid devices extend beyond energy conversion. We won't talk about that here. For anyone who's interested, we are having another RSC symposium on this very topic, where you get to hear about other types of biohybrid approaches to sustainable energy conversion, where I'll be chairing along with Nicola Plumeret and Owen Reisner. So please join us there. Poster abstracts are due soon, so though we are quite flexible, so don't let that put you off if you want to submit, but do please join us there. I hope to see you there. And with that, I'd just like to thank everyone, my group members who are absolutely amazing. Some of them are joining me here today, you cannot see. And also, of course, Professor Stephen Harding, who is so generous with his time and his introduction and with this award. Extremely honored, all my collaborators, because it's an extremely dynamic and interdisciplinary field. And I'd like to thank you for your attention. So if there's any questions that you like to ask, I'd be happy to address them.
Okay, thank you, Jenny, for your very interesting talk. I can see some questions in the chat box. So first, the first one is this the inkjet kind of 3D printing or some other 3D printing method?
It's similar. It's like a hybrid between inkjet and the extrusion base, but it's not quite completely inkjet printing. But if you want details, actually, it's up on Chem Ref Bio, so I could put you in contact. You can have a look at the precise details of the printing method, but it's a hybrid printing method.
Okay. I think the second question is almost the same, similar as also concerning your 3D printing. Are you 3D printing a paste containing ITO particles?
Yeah, it's actually not a paste. It's a ink, that what they call ink, and the ink composition is really important. Actually, you need to make it so that it's got some volatile and non-volatile solvents co-mixed inside, so that it holds together, but then also it evaporates fast enough so that it can form a structure. So yeah, so I hope that answers your question. It's more like an ink, not a paste. So yeah, because I think you're thinking of extrusion printing.
Yeah. I should say the first question is from Natropol, the second question from Vincent. Okay, and I also have a question myself. Yeah, you have used PS2 and hydrogenase as both water oxidation and proton reduction system, and bind them together to construct a PEC cell. I want to know, is it possible for such a PEC cell to overall splitting water at 0 bias?
Yes. We have made that happen. Yeah, so basically using a tandem biohybrid system. So we have to introduce an artificial light absorber in there, because we need to basically change the energetics of the electrons exiting the photosystem too. And one way of doing that is to basically excite those electrons again. And so we use dyes, we use semiconductors, and basically the dye one was extremely successful. But I'm sure also now I think Erwin Rice's group also developed some other devices where they coupled it to a perovskite photovoltaic cell as well. So I think that there's many different strategies you can go into making a bias-free.
Okay. Yeah.